Yeah.
We went to elevator.
So. Especially if you're struggling too.
There's this decaf. Decaf's in my right hand. So my younger daughter, Haley, works in a restaurant in Spokane. And it's a really nice restaurant. And so she's gotten used to moving things around. So she comes and cooks for us. She'll bring out the food stuff, start tossing things down. She's a runner, so she's like brings food out to people. She loves it. Yeah, she's doing really well. Yeah, she's thinking she did medicine in law school. She got into it, started to really work with professors. Decided that she's going to go to grad school. She's going to PhD in criminology. She had a fellowship to research this summer and through the next academic year. So she's really excited about it. She's going into her senior year. All right? Yes, it is here.
That's not really loud.
Jordan is she's working.
I'm Arthur Glasgow, I'm to the left, I'm the Alexander Hay who's stepping in for Andrew tonight, because you know what that means in the old time. Anyway, I will be starting the ceremonies in about 5 minutes. And so if any of you feel like a bathroom break or another food grab would be in your best interest, the bathroom is around the corner, which is there, and we will get going shortly.
So this is Joseph from Meyer. It's a guy, he graduated from UP in 2008. And that's from Brazil. So he's
especially like diseases using this
yeah, I remember interview with friends. He's in first so he was hospitalized. I think so. Andrew was hospitalized. His last name is anyway, he was coming back. So the students are all it's become a labor of love. This weekend, my powers is there was Ryobi battery packets. Yeah. I hate it because I grew up doing so much. But now that I can get all these tools, it's like playtime.
Basically,
Yeah, caffeine has like
and then methylate it and make caffeine. Different class. And I started looking at it. The instructor was the student who's sulfate is calculates DNA so well. I read all the stuff and we never I didn't want to stay.
DMSO.
So it's like sulfuric acid. It's an extremely sweet, but it's so difficult
All right. Thank you, everybody. I hope we're feeling settled into the evening. I'd like to start by inviting Gary Davis to come up, and he's going to present the NWSP Award this evening. Oh, I should say masks. My teacher reads like for the policy. If you're eating, you can stay unmasked, but the college rules, if you're indoors and not eating, you should be masked.
Good evening, everyone. It's my distinct pleasure tonight to be able to present one of the outstanding awards for high school chemistry. I'll start by saying that looking out in the area here, it's wonderful to see many young faces. That's because many of us who have not so young faces need to pass the baton to those of you who are younger at the American Chemical Society local section. The award that I judged, there were two special awards for high school chemistry students, and the one that I judged, I awarded to Audrey Dunn. Just a little background, Audrey is a grade 9 student at Sunset High School. And Audrey, I have to tell you that a number of decades ago, I graduated from Newton High School. So a fierce rival of Sunset High School. And that number of decades ago, Sunset High School was the new school in the district. So they got all the fancy stuff and we just got the barn. In terms of looking through a number of the abstracts for the potential awardees, several things caught my eye about Audrey's abstract. She presented a rather broad overview of the project and her rationalization for carrying this project out. So she combined an interest in environmental sustainability, the chemical health of soils, and how that related to the application of fertilizers, and the use, this especially caught my eye, of several analytical techniques to analyze results and come up with a nice story. And I think that it goes without saying that with the current situation of climate change and the overuse of soils that are being overstressed in commercial farming, this is a topic of current interest. So the title of her project was the determination of low-cost tools for novel health characterization in contaminant remediation methods. And I kind of reinterpret that to say, how do you do research on a shoestring without having very expensive instruments. In her abstract, she clearly answered the question of why. Why is this research interesting? And many of you have looked at her poster over here. I feel like you were cornered over here by many people. You didn't get a chance to even walk around. Everybody was interested in looking at the poster. And so you'll see that many of you who look at her poster will know that her project concerned, for example, the addition of fertilizers in the form of aluminum nitrates, which introduce the nitrogen content of the soil that's needed, but left behind residual aluminum, which kills off some of the desirable microbes. So that was the why. And as I say, I was also attracted in her abstract to the variety of techniques, analytical techniques she proposed or that she used to carry out her project. Amongst those were thin layer chromatography, and something you can do on a shoestring budget. Doesn't cost much, but gives you a wealth of information. She also used a technique which was a variation of something called laser-induced breakdown spectroscopy. That's not something you want to try and do real fast many times over. That technique is used to detect particular metals, and instead she used a technique which again caught my eye in particular because of an experiment that we have designed at Portland Community College for our undergraduate labs. Using the technique just uses a handheld laser and basically looks at a diffraction rating from a compact disc in Audrey's case, and then uses this technique to, and I won't go through the details, you can look at her poster there, to identify the presence of a particular metal in the soils. She did this in collaboration with Emmanuel Abdul at Portland State University. Was one of the other techniques that she did employ to look at the fatty acid content in human materials. So that was one of the ways you can judge the health of the soils by looking at the fatty acid content. And she used NMR spectroscopy to do that. In conjunction with Emmanuel Abdul at Portland State, since that's the nominal cost of the experiment. I have to, as I read her abstract and looked at her biography, it was in grade 9. I could spell NMR, but I had no idea what it is. So I would like to present this award tonight to Audrey Doss. Congratulations, Audrey. And Audrey is following not exactly a long line, because it's just one predecessor for her. Her sibling won this award some years ago, and so the whole family is following along in the same patterns here. So congratulations to Audrey.
Thanks, Gary, and congratulations, Audrey. So I am charged with presenting our college scholarship winners with their certificates and their scholarship today and introducing them to you. And so, Juan, if you'd like to come, it'll have 3 present today. Unfortunately, 2 couldn't make it, but allow me to introduce Juan Hernandez. I'm sorry I haven't met you before. Congratulations. Anyway, is a student at Willamette University. He's from Kaiser, Oregon originally, and he is the recipient of the James G. Anderson Scholarship. The scholarship committee has noted that along with non-chemistry, because as he said, it allowed him to better understand the world. And recently, he's also found it enjoyable to teach others what he's learned. Juan is one of the top chemistry students at Willamette. He's also a creative one. When Juan took an organic chemistry course recently, he was asked to produce a podcast, and one he did on the role of PDTA in everyday life. That podcast was actually selected for broadcast on a local radio station, KMUZ-FM. And in addition, he's been described as being extremely responsible, dedicated, and a great role model. Juan was a general chemistry lab TA during the period of hybrid teaching during COVID, and I think as many of us know, we relied very heavily on our student assistants during that period. Juan shouldered that extra responsibility with aplomb, as one of his instructors. He has a calm, quiet presence, and was a recommender who could be confident that Willamette students were good hands whenever Juan was present. I'm delighted to present the James G. Anderson Scholarship to Juan, supporting his continued studies in chemistry and in celebration of his achievements to date. And I showed you the wrong, this is exactly like the Oscars.
No, you're not going to get slapped.
Oh, wait, this one, I did it. I set up this one. Yeah, congratulations.
Okay, just want to make sure it works. Well, I do appreciate the award, and one thing that surprised me about this was, I think I was sitting at my home and I got an email from one of my advisors like, hey, we nominated you to apply for the scholarship. I'm like, really? Because honestly, like, this year's been so crazy, these past years have been so crazy. So being able to be here and being able to get this has been really reassuring to my future. And I just hope that one day I can be able to support other students too, because I'm going to be a teacher. So thank you everyone, and yeah.
So our next scholarship winner is Emma. Oh, congratulations. So Emma is the winner of the Harold Zay Scholarship. Emma is a student at the University of Portland, and she hails from Portland originally. It was in Emma's junior year in high school that she realized her genuine interest for chemistry. And it was due to that atoms and molecules are the building blocks of everything in the world. And she made up her mind to major in chemistry in college. And despite the stereotypic depiction of organic chemistry as being stressful and difficult class, Emma found it intuitive and even fun to draw out mechanisms and figure out the steps to synthetic problems. You have to be writing back to Emma joined a research group at the University of Portland after her first year, which is apparently quite unusual. And that was in the summer of 2020, because I think we all recognize one of the difficult years to actually be doing research anywhere. And her research advisor notes that in her first summer, it was clear that Emma was light years ahead of her peers in her ability to learn and solve problems independently. She undertook the computational organic chemistry project without ever having taken organic or physical chemistry. And she essentially taught herself the fundamentals of those subjects in the first summer of research. And that summer has been followed by a second research internship more recently using infrared spectroscopy to identify microplastics in stormwater runoff. It's a pleasure to provide Emma with Harold Zay Award in recognition of her outstanding work at the University of Portland and the great promise that she shows for future work in chemistry.
Congratulations. My name is Emma Morrow. I'm a junior, and basically how it happened was kind of a similar thing to Juan. Was I basically was nominated for this scholarship, and I was like, well, okay. But yeah, I really appreciate this opportunity. These past couple of years have been very difficult, and I really still, I've always really loved chemistry and being able to do it even though it's hard, and finding my love for organic chemistry. I'm just really thankful for this opportunity. My plans are to basically go to graduate school and pursue research in computational, as we discussed earlier, or I'm still trying to decide what I could do organic synthesis in the lab as well. So I can't really decide because I like both, because I've had experiences for research in both. So yeah, thank you so much for this opportunity.
Unfortunately, our third scholarship winner, James Sheldrake, who's been given the Richard T. Van Zandt Award, is currently, this is unfortunate, she's staying for studying abroad. And she's from Lewis and Clark College, and originally hails from Seattle. And what I'm told from the scholarship committee is that in the high school chemistry class, Megan's group spent the whole class period figuring out the periodic table. And she discovered the simple joy of asking questions, so that when the time came to choose a major in college, it was chemistry, because of that curiosity. She wants to know not just how things work in our world, but why they work that way. In her quantum chemistry class, Megan undertook an ambitious final project of modeling the heat group in order to study its conformational change upon oxygen binding. Her professor in that class was impressed by her ability to adapt and overcome obstacles and learn from the project, and by her ability to communicate her results as well as her ability to connect her work to topics that were covered during the course of the semester. Her 2021 summer research was characterized by critical thinking and coming up with solutions on the fly against many obstacles in research, and this was coupled with her persistence and dedication that is a common trait that was spelled out in all of her recommendation letters. And so, in absentia, it is a pleasure to honor Megan with this award, and maybe Anne can help me see that this gets and we start with, well, Anne, let me also say that I think it's really wonderful that the faculty of these students have been able to provide them with the opportunities to let them grow as chemists under the circumstances of the past 2 years. So the next two scholarship winners are being awarded the Tom Dunn and James Curry scholarships, and I think many of you knew Tom and Jim very well. Tom, for me, was a close colleague and mentor, and this is a really meaningful award to me because I know both of them care deeply about issues of equity in chemistry and social justice, and I think they'd be particularly pleased to know that scholarships are going out in their name to students who come from typically marginalized groups in support of their careers in chemistry. And these are sophomores who have already made a very impressive start to their academic careers. Melissa Duncan is if you come up. So welcome Melissa Duncan, who comes to us from Willamette University. She's a sophomore in chemistry on the biochemistry track and is pursuing minors in biology and math. That's a lot. So Melissa's key achievement is participation in summer research after her first year of Willamette. Melissa worked on a nucleic acid folding problem with the long-term goal of developing fluorescent sensors that detect non-coding RNA. Again, there's impressive quality that our students are doing work they never had coursework in and learning it on well, because you never had organic or biochemistry prior to that summer. Yet it appears that you did fantastic work. You were invited to present your work at the Murdoch College Research Conference this past fall. And she's described by one of her professors as a self-disciplined and independent learner with a strong passion for applied mathematics and science. Her research advisor applauds Melissa's talent, maturity, persistence, and independence. And so it's a pleasure to support Melissa with this scholarship, and we congratulate her for her achievements so far, and look forward to many great things to come.
It was like 3 minutes of how country impacted us.
Uh-huh.
Okay. I think I read it wrong way, but I'm still going to go with it, partially. So my name is Melissa Duncan. As was said, I'm a sophomore at Lamb University, majoring in biochemistry and minoring in mathematics and biology. I intend in my future to pursue an MD PhD with a PhD in biochemistry and hopefully become a researcher in medicine. My current goals this summer and next summer are to continue my research with Professor Battle, my research advisor, and hopefully get a publication by the budget this year, next school year. Personally, I didn't actually have any chemistry background until I came to Lynn University. My high school didn't really provide those opportunities and labs for frequently not exactly looked at as great. So I was really excited to come to university and have the opportunity to actually experience labs. And I remember my first lab, I was so excited and so nervous that I wasn't able to actually properly measure out reagents for an experiment. That I really just, I'm grateful for all of the professors and all my peers who supported me in getting me to where I am today, including Professor Williamson, the professor of that lab that taught, as well as my research advisor, Professor Battle, for taking me on this the first year, and of course, the American Chemical Society for their support, as well as for the support of other representative minorities in chemistry. So thank you, and thank you all for listening.
And unfortunately, our other Dunn Curry Scholarship winner can't be here this evening. Marnie Osmond is also a sophomore at Willamette University, and she is a sophomore in chemistry and data science. So, an ambitious group at Willamette. One of Marnie's recommendation writers describes Marnie as ambitious, organized, and self-motivated. She will be the first chemistry major to also double major in data science. And in the summer of 2021, Marnie was part of a collaborative NIH research project between Willamette and Washington State that tested the effectiveness of ion channel inhibitors in therapy. As one letter writer wrote, she was traveling to another state for the summer during COVID times, and it seemed to be a fairly daunting undertaking. But Marnie managed to solve problems, made real progress on the project, and colleagues at both Washington State and Willamette are impressed by Marnie's leadership role in being a driving force that advances the collaboration. Marnie's project was also selected by Willamette to be presented at the regional Murdoch College Science Research Conference. And Marnie's excellence in research and science communication has been a great source of pride for her research team. And so I'm pleased to present this award, which has been mailed already, in absentia in honor of her most promising start in chemistry. Three of our 5 scholarship winners. Great things happening in sales. So, with that, I believe I hand the floor to Dave Reinhold, who will introduce our speakers.
It's almost 8 o'clock. I have this long introduction for Joel. I'm not going to use it. But I want to give him enough time to give his talk. So I'll just mention that Joel showed up. I taught organic chemistry for 35 years. I don't think any other student ever appeared in my class as a senior. People don't take organic chemistry for fun in general. And they take it because they have to. And if they're taking it as a senior, it's probably because they failed, or they were just scared shitless, or for some other reason they put it off. None of this turned out to be true. Joel was a great student. It was lovely to have him in class. He graduated from Juilliard as a major in peace and conflict studies, which probably doesn't exist in many other places, and also environmental studies. He went off to Guatemala for several years doing social justice work there, came back, went to environmental toxicology school at Duke, went off to a postdoc. I hired him back at Duke. This is unusual as well, I think. It is most of the places. And he's been there ever since. It's a pleasure and an honor to introduce Joel Meyer.
Thanks so much, Dave. I practice with this, but not with a mask, so I'm going to try to attach it to my mask, and if it does not work well, just let me know, and I'll attach it elsewhere. What I think? Is that okay?
Yes.
Fantastic. Great. So, thank you, Dave. Thank you to the local section of ACS in Portland for inviting me, and thank you all for listening. As mentioned, I'm at the School of the Environment at Duke University. I'm an environmental toxicologist, and hopefully I will make a case for why that might be interesting to those who are mostly panelists, I believe. I want to start out with acknowledging a lot of incredible collaborators and folks that do a lot of this work. I can't show them all, this is more than 10 years worth of work. I'm highlighting some of the people that were most critical. PhD student Gino Yao, postdocs Laura Meyer, Daniel Mello, and Lou Wang, and two really key collaborators at Duke's Civil and Environmental Engineering Department, Helen Shi Chen and Mark Wisner, were critical to the work we did. There were many others, but they played a really large role, and we had funding from NSF, EPA, and NIH during this time. And I do have a picture of Dave up here because he's opened multiple doors for me. First of all, to excitement about and enjoyment of organic chemistry as a senior. Second of all, I think it was probably his letter that helped me among the Thursday to graduate school, and of course, why I'm here. So thanks again for that opportunity. This is an outline of what I'm going to talk about. I'm going to start out a little bit with what is environmental toxicology, and why might an environmental toxicologist be interested in nanomaterials, and then I'll introduce nanomaterials themselves a little bit, and why we got worried about them. Then I'll talk about some of the tests we did on nanomaterials along the way, in particular silver nanoparticles, is what we spent most of our time studying. So, environmental toxicology. Why should we be interested in this? There are a variety of ways of thinking about this. One of them, in my mind, is the fact that as we've gotten rid of many of the things that used to cause people to die early, things like infectious disease, lack of sanitation, and we're living longer and dying of chronic diseases, we're running into the problem that many of those chronic diseases are not well explained by genetics. Much of what's driving this disease is things other than genetics, so environmental factors of various sorts, not just pollution, of course, but including pollution. And there was a study that was an entire issue of the Lancet Journal about 4 years ago, based on 2015 data. And it really shocked me, which is embarrassing because this is what I do, I should know this. But they asked if we looked at these various factors, AIDS, malaria, pollution, tuberculosis, war, which ones are most important? Which ones cause the most loss of life globally? And I like to ask this question to students, but again, I would not have known the answer to this until this came out. The answer is that pollution causes 15 times more deaths globally than war, and 3 times as many as AIDS, malaria, and tuberculosis put together. So it's a major effect on health. And we'd like to understand this as well as we can. So that's one big motivation. This is just a reference if anybody's interested. It's the whole issue of the Lancet. Another way to think about it, and one that maybe is particularly interesting I looked at this crowd, is that environmental toxicology lets us balance the benefits of advances we make in chemical engineering and other forms of engineering with potential risks associated with those. So how do we appropriately balance those two? And I've got a bunch of sort of famous examples of things here that are useful in many ways. So pesticides, detergents, fluorinated refrigerants. Lead is actually a useful drug. Lead has useful properties. But all of these led to very famous, well-known examples of environmental and human health problems, because we just weren't prepared for some of the potential toxic effects of them. So the toxicologist tries to understand these, hopefully, before they occur, so that we can avoid this sorts of problems. We avoid the unintended consequences of soil spraying, of eutrophication in lakes, of ozone hole, of children born with deformed limbs, of lead poisoning. And about 20, 25 years ago, people began making a lot of these nanomaterials. They've been making them for longer than that. And the question was, can we play a role in trying to avoid unintended consequences associated with the development of these interesting materials? So around 2000 is when this really took off. Again, people had begun engineering nanomaterials a few decades before that, but it was really taking off commercially around that time. This is a quote that kind of captures some of that. Is it, and I refer to this quote, carbon nanotubes, is it the next best thing to sliced bread, or is it the next asbestos? And the reason people wondered about that, this is a picture of asbestos, long, inorganic, tube-like structures that can get stuck in lungs, and because they cannot be broken down by your body attempts to break them down, causes inflammation, ultimately causing various kinds of lung cancer, lung disease, including cancer. And if you look at a carbon nanotube, it has a very similar structure. It's carbon, but it's also extremely persistent, and there was concern that turned out to be legitimate that it might have a similar effect. So, do we need to be worried about this, and what about other nanomaterials? Because it turns out that smart people were coming up with a ton of different kinds of nanomaterials. I haven't told you what they are yet. You may know this already. So I'll be fairly brief, but nanomaterials are typically defined as materials that are between 1 and 100 nanometers in at least one dimension. So maybe more than one, for example, along 2. At least one of the dimensions is between 1 and 100 nanometers. And for a sense of scale, this is so between sort of the width of the DNA helix up to slightly smaller than a virus is the size we're talking about as nanomaterials. People take some other definitions. Sometimes these are also defined by the fact that their properties change with size, which is not true of larger bulk materials, generally speaking. They come into 3 categories. Natural, there's lots of naturally produced nanomaterials out there. Incidental ones, things like air pollution. And then engineered ones, ones that I'm going to create this thing because I think they'll use it. And that's really what got people thinking a lot about possible toxic side effects. Although in retrospect, we already knew a lot about the toxic effects of some forms of air pollution. It's referred to as ultrafine air pollution. That's less than nanometer size. So we did actually know about that. Now they do really cool things and really interesting things. This is a picture of a bacterial cell being killed by solar nanoparticles. And as I mentioned, I spent most of my time talking about solar nanoparticles. They're really good for that purpose. Again, these are often materials whose properties change with size within that 1 to 100 roughly nanometer size range. And that can be their electrical conductivity, that can be a variety of other properties. This is a commonly used example. These are cadmium-selenium quantum dots. And just by varying the size of those dots between 2 and 8 nanometers, you get a change in the emission spectrum upon radiation with high-UV light. It's sort of a simple illustration of the fact that these properties change. There's no change in the makeup, just the size of those particles. And this is a little bit dated now, but it just makes the point that people are inventing and engineering and creating lots and lots of these interesting particles. So this sort of led to this question of, nanomaterials, are they bad for the environment? Should we worry about these? Or are they good for the environment? And there's certainly a strong case to be made for them to be good for the environment as well. They're used to clean up contaminated groundwater, for example. They're used to build very light, very strong materials such as cars, so that they can be more fuel efficient and at the same time more safe. They're used in batteries and solar panels. So there's a lot of ways in which they could be good for the environment. Or are they bad for the environment? I talked about this one. We certainly know that very fine air pollution is terrible for human health. There was a lot of concern, for example, people put a lot of nanomaterials in sunscreen. If you think about the white sunscreen that you used to wear, you don't see it around very much. That was usually titanium oxide. If you make that nano size, it's clear. So now you can be protected from the sun without having a white face. So lots of people preferred that, but there was again this concern: is this going to be a problem for health? So that's sort of where we came in, and we were interested in beginning to test possible toxic effects of some of these materials. To do that, we did most of our work either in cell culture or else using this beautiful, elegant worm called C. elegans. Some people tell me that it's just serving after dinner, so I apologize if that's the reason. I think it's beautiful and elegant, but not everyone is the same age. It's about a 1 millimeter long, non-parasitic worm. It's transparent, as you can see. Its biology has been really well studied by geneticists and developmental biologists. So if you want to ask how's the normal biology of this organism affected by exposure, it's handy because we already have this large database of knowledge of how does this organism normally behave. There are mutants available, so if you want to ask how this mutation in its gene affect the worm, it's pretty easy to get a hold of those. There are transgenics. This is a worm in which GFP, mucin protein, has been expressed just in neurons, so we can ask about, does a certain exposure cause neurotoxicity or neurodegeneration? And generally speaking, there's pretty good genetic conservation with higher organisms like us, or like what other species we might be interested in. Obviously, there's some major physiological differences, but at the genetic level, there's pretty good conservation. And if you don't believe me on that, you can believe the guy with the really impressive mustache. All the worms in my hand, and much more than you this world are. So clearly, a pretty model for that. Okay. One of the things we'll often do as a first step, just to say how toxic is something, is we'll take worms and take them at their first larval stage, they go from 4 larval stages to adulthood, and grow them in the presence of some stressor, and ask how much is that growth retarded. An example that is shown here. So with these silver nanoparticles under control conditions, this is the size. Each dot here is a worm. And with increasing amounts of silver nanoparticles, you can see they grow less. Right? And the way we typically quantify that is we say, okay, how large are the control worms? Here's the average size. And then we'll say, what's half of that? And what is the concentration at which we get a 50% reduction, in this case, in growth? That's often called the effective concentration 50. And it can be for growth, it can be for lots of other outcomes. What's nice about that is that gives us a single number, the EC50, it captures all of that information. Not actually all of it, but it summarizes it. This is roughly how much it takes to inhibit growth pathway. And what's nice about worms is you can do that with a whole bunch of chemicals and compare them, or a whole bunch of nanoparticles and compare them. And this is why we got to silver nanoparticles. So if we look here, here's a whole bunch of different nanoparticles that we tested for EC50s. So how much was growth inhibited, in this case 72 hours, after 72 hours of exposure. And what you can see is that the silver ones, generally speaking, were relatively toxic. Down here is C60, sometimes called buckyball, one of the fullerenes, really non-toxic in these assays. It does almost nothing. And you can see there's roughly a 3 order of magnitude difference in the toxicity, the ability of these different particles to inhibit growth. So that was one of the things that said to us, let's pay more attention to these silver nanoparticles. Maybe not surprising in retrospect, the goal of silver nanoparticles, or at least the most common use, is to kill microbes. So they are, in fact, designed to kill things. So that was one reason that we decided to spend a lot of time on silver nanoparticles. Our initial screening, plus other people's initial screening, said yes, these look pretty toxic as they go. Relatively high volume use compared to other nanoparticles. Some are higher, but it's relatively high. Also, they're currently using personal care products, so this is just a band-aid. But because they're antimicrobial, they're incorporated into all sorts of things. So you're guaranteeing human exposure. Sometimes in the context of a cotton or scrape. Also your socks, so they don't smell. And then finally, because of often using clothing and all other uses, it will end up in sewage. And so sewage is often partially treated, but not always fully. That can get out to the environment, affect ecological systems. The sewage may also be applied to land where it might be to improve agriculture, for example. And so, ideally, we're not going to have something that'll be killing microbes and apply to agriculture. So these were some of the concerns that we started with when we first decided silver nanoparticles. Because we were interested in this entry into the environment, we decided to spend some time asking not just what are the toxic effects of the particles in water, basically in the laboratory with these worms, but in more realistic environmental situations, because that's not a common environmental situation. And when we thought about it, we knew that these particles would be moving around. Yes, they would go to sewage treatment plants, they would move all over the place. Some of them might end up in rivers, some of them might end up in estuaries of the ocean, some of them might end up in soil environments. And so we expected them to encounter sulfide, which is really common in certain sediment areas of ecosystems. Chloride ions, especially where there's significant salinity in the water, and natural organic matter present in most water bodies. So we decided to take a look at all of these. And one of the first things we found, so here what we're doing is actually not testing toxicity, but just testing the size of the nanoparticles in solution over time. So if you take these particles and put them in just pure water, these particular particles usually stay in suspension. And you can see that in water here, at least over 24 hours. There's no change in what's referred to as the hydrodynamic size. That's just telling you that they're not agglomerating, not sticking together and creating larger particles over time. But if you put them in what we call K plus beads, which is basically a potassium and sodium chloride mixture that we put the worms in, there's a lot of chloride. They quickly aggregate. In fact, they quickly become non-nano in their size. And I'm not showing you this data, but they also become much, much less toxic. So in pure water, they're pretty toxic. They agglomerate. In water with not much chloride, they do agglomerate and they become much less toxic. And that was true for a variety of different types of silver nanoparticles. I'm not going to talk a huge amount about the differences here, but we look at different sizes and different coatings. And this is a pretty common theme. We also looked at the presence of sulfur, and this was actually a really interesting, this was work by Candy. This is a really interesting collaboration with about 5 different labs using 5 different organisms. And we all got the same result. In our case, it was worms, people, 2 or 3 species of fish, some marine invertebrates. And what we found is that we take a dose response, so increasing concentrations of silver, and in this case, with no sulfur added, you get an increase in lethality. So that's just a dose response for lethality. If you add a little sulfur, not much changes. If you add more sulfur, we start to see a decrease in toxicity. So now there's no lethality at 15 parts per million, whereas it was at lower amounts of sulfur. And if you go up to a high level of sulfur, it really pretty much takes away the toxicity altogether. And this, again, was seen in every organism that the different laboratories tested. That's encouraging because most silver nanomaterials that we're worried about how they're going to go out in the environment, we would expect them to encounter sulfur-containing material. So that was a little encouraging. And then finally, Candy also looked at natural organic matter. It turns out there are standardized forms of natural organic matter that you can purchase. 2 of them are from Pony Lake and Suwannee River. And they vary mostly in nitrogen and sulfur content. And what she found is that as you increase the concentration of natural organic matter, the toxicity, again, percent mortality, comes down, much more so for the Pony Lake and the Suwannee River. But again, this indicated that the presence of these natural ligands that would normally be found in most natural water bodies, not all, significantly decreased toxicity. So that was also encouraging. And to summarize this sort of an ecotoxicology, this sort of ecological set of work, what we found is that everything we looked at seemed to decrease toxicity compared to what we see in laboratory conditions. However, we didn't test everything that could be tested, and I want to leave that as a caveat. There are a few nanoparticles, I don't think this is true of silver, but for example, that titanium dioxide is phototoxic. It absorbs sunlight, it can actually transfer the energy of those photons to oxygen and create reactive oxygen species in So that causes a really large increase in toxicity of the titanium dioxide nanoparticles, not the silver ones. We didn't look at more acid conditions, and that is one that I worry about with the silver nanoparticles, as I'll show you in a little bit. More acidity means more dissolution of silver, and in our hands, most of the toxicity of these particles is actually driven by distribution. So that could be. What we really spent most of our time on, though, was trying to understand how the particles were being toxic. And that's what I'm going to spend most of the rest of the talk on. So not just are they toxic and what environmental conditions might change that, but how are they being toxic? And the reason we really wanted to know this is we already have a literature on the toxicity of silver. The question was really, are silver nanoparticles different than silver? We know that they have different properties in terms of physical chemical properties that can be useful in industry. We didn't really know if they could be different in terms of toxicity. And that was generally speaking the big concern we had about nanomaterials in general. If they're different in terms of their industrial applications, are they different in terms of toxicology, which means we're starting sort of from scratch in terms of understanding their effects. Okay, so we'll move on to that now. You've seen a little bit of this data, but this is looking at a variety of silver nanoparticles as well as just silver nitrate. We typically use silver nitrate as our dissolved silver ion control. And ask what again is the EC50. And you can see there's even within the silver nanoparticles, there's at least a 2 order of magnitude range. Some are much more toxic than others in terms of growth inhibition. So why? What's different? Is it the size? Some of them have different coatings. Is the coating? These are all spheres. I'm not even going to talk about other shapes, but there are other shapes out there. What's really responsible? And the reason that matters is because people make lots of these of different sizes and shapes and coatings. And if all we can do is test every single one at a time, toxicologists can't keep up. There will be many more produced than we can possibly test. On the other hand, we come up with some generalizable rules about this is what controls toxicity, then that at least gives us a head start on getting a handle on possible toxicity of new products. So mechanistic understanding of what's maybe driving toxic effects is needed for treatment. We started out with a few different hypotheses about how they might be causing toxicity. One was oxidative stress, and this was really derivative of the air pollution research. So air pollution-related small particles typically permit redox reactions on their surface, and that causes oxidative stress. And there was evidence that was in vitro in water that you could get a similar thing to happen with silver nanoparticles. And that would be a particle-specific effect. Ions are not going to do that, right? You have to have the particle that has surface chemistry. Another idea was that metal ion toxicity after the particles or as they dissolve would drive toxicity. So in that case, the particle would really just be affected to get the ions to be dissolving somewhere. In both of these cases, we predict that smaller would be more toxic, right? So with surface chemistry, smaller particles have a higher ratio of surface area per amount of mass. You should have more surface chemistry. Because dissolution, you should have faster dissolution. So in both cases, we expected smaller will be more toxic. And so to get this, these are some of the approaches we took. We tried to correlate the toxicity with the size and with the rate of dissolution of these particles. We also went to our worm model and tested mutants. So they're strains of worms carrying mutations that sensitize them to specific types of stress, to ask whether those specific types of stress were driving toxicity. And then we use a chemical or pharmacological rescue, so antioxidants that would presumably, for example, block oxidative stress, if that's what was driving toxicity. And I'll walk you through a little bit of the data from each of these. So the first thing was just, is it straight up size? And the answer is no. So here we're looking at the average monomer diameter of a bone coated 5 nanometer silver nanoparticles, silver nitrate, PVP coated 38 nanometer, bone arabic again 22, PVP8, citrate coated or functionalized 7 nanometer. So we have a range of sizes. We including 3 pretty small ones that are all roughly in the same range, 5 to 8 nanometers. And there's really no correlation between that EC50 value, that growth inhibition, and size. But if you plot the fraction dissolved versus EC50, there's a really nice correlation. And so what we think is happening here is that it's not just the size controlling rate of dissolution, it's also those coatings. And so you have to take both of those into account. And if you just functionally measure how fast they dissolve, that gives you a really good predictor of how toxic they are. So that was supportive of it being an ion effect, and it really driven by dissolution. We tested that in some other ways. So here we're looking at using some of the same silver nanoparticles and silver ions, and we're testing in what's called the wild type strain. This is just a normal worm with no mutations that we are aware of. And we defined the level of exposure to each of those that was sort of mildly toxic, that had anything with just a plus. And then we tested 2 mutants that are metal ion sensitive, and that's because they both failed to produce 2 different small proteins that have lots of sulfide groups that accumulate ions. And so they should be sensitive to metal ions. And in fact, we knew that was true. If you just expose them to metals in solution, they're much more sensitive than wild type. And then we also exposed 2 mutants. One is a superoxide dismutase, the other is actually an electron transport chain mutation that are very sensitive to oxidative stress. The idea here was, well, okay, if these are much more sensitive, then it's probably the metal ions. These are much more sensitive, then it might also be oxidative stress. And what we found was that universally, all of the metal ion sensitive mutants were sensitive to all nanoparticles. So again, and they were also sensitive to the silver nitrate. Again, supporting this ion idea. When we looked at the oxidative stress mutants, it's a mixed story. So they were not more sensitive to silver ion. That's what we expected. They were more sensitive to 3 of them, and actually in particular this small citrate functionalized silver nanoparticle. They were quite a bit more sensitive to that one. So that made us think, well, okay, maybe in this case, the oxidative stress mechanism is important. And it's not an indirect dissolution oxidative stress because the sulfur nitrate by itself doesn't show the sensitivity. I don't know if you remember this, but that citrate-coated particle was the least toxic of the ones we tested. And so what we ended up thinking based on this is that it seems like the metal ion dissolution drives toxicity unless it's really slow. And if it's really slow, then the redox activity begins to matter. And we tested that one other way, which was using an acetylcysteine, which is an antioxidant that's a defined precursor, but it's also an ion chelator by itself. So it sort of takes both of those effects. And then Trolox, which is a more water-soluble version of vitamin E, is an antioxidant, but doesn't really have strong chelation capacity. So it should be an antioxidant without having much ability to chelate the ions and take those away. And so if we look here, you can see with just silver nitrate, as expected, the acetylcysteine residues, so they're larger after exposure than without the acetylcysteine. And the Trolox have no effects, as we also expect with the silver nitrate. We got a very similar result with the PVP-coated 8 nanometer particles. Also, the PVP-coated 38 nanometer. However, again, with the citrate 7 in particular, we do see some rescue with the Trolox. We see rescue with the acetylcysteine, but also with the Trolox. Again, supporting that this particular particle does have some particle-specific, potentially oxidative stress effects. With the others that we tested, the Trolox either had no effect or a very mild effect. So this together, this sort of different lines of evidence made us think most of what's going on is a solution. There is, however, in particular for the citrate-coated particle, a role for a particle-specific role. So we know dissolution is important for all particles that we tested, and oxidative stress appears to be important for some. If there's a particle-specific effect, then that raises a set of additional questions, as usually happens in science. Does that mean that the particles themselves are being taken up, or are they just acting at the surface and giving a big up there is uptake, we'll correlate the sites of damage. What are intracellular targets? Where is it going? And how does uptake occur? So here we took advantage of the clear nature of the worm, the transparent nature of the worm, and using a form of dark field microscopy, we're able to see the nanoparticles ingested. This is the pharynx, this is the gut. And if you expose them for long enough, you would get transferred past the gut into other tissues and even into the eggs. So they were actually being taken up and transferred into the next generation. The majority went through the gut and was excreted. This is potentially not that surprising for an organism that lives in sediments, but some was taken up and did have an effect. We looked a little closer with transmission electron microscopy, and this is looking at the gut lumen of the worm, so you can surrounded by gonadal and muscle and epithelial cells. And so this is what it should normally look like in terms of the tissue structure. And after silver nitrate, we got swelling of the intestinal epitheliums and intestinal cellular damage, which was not at all surprising to us, but that's sort of what we expected to see. When we looked at silver nanoparticles, interestingly, we actually saw damage, and again, we're intentionally using those citrate-coated particles that seem to have a particle-specific effect here. We get damage that goes further into the body, past the intestinal epithelium, and actually causes no damage at all to what's referred to as the brush border of that intestinal epithelium, which really suggests that the particles are getting in and causing damage at these more internal sites. So this suggested maybe a role for endocytosis. It's hard to imagine how these particles would be getting in without endocytosis. I want to mention that we saw some mitochondrial swelling, and that's because this is some of the data that led us to then later wonder whether mitochondria were a target of these particles. We saw a little bit of location of the swelling, and again, this sort of hinted at maybe a nanoparticle specific effect for this one type of particle. So now we know the solution matters, oxidative stress may matter for this one particle type, and we do get some crossing of cell membranes of these particles. How does it occur? And we have the hypothesis that it might be endocytosis, as I already mentioned. We tested it using some of the same techniques, endocytosis mutants that couldn't perform endocytosis, pharmacological blockers of endocytosis. And I'm not going to show you the data for a couple of reasons. I will mention that most of this work was done by Laura Bauer. But the main reason I'm not going to show you is other people scooped us on this one, we just did the punch, published it, and furthermore had a way better schematic than we do, so I'm going to show you their schematic instead of ours. So basically what people found, and this turns out to be true for many metal nanoparticles, not surprisingly, they're taken up by this process of endocytosis, so they're not just sort of diffusing across the cell membrane, they're actually actively engulfed, and then those vesicles move to lysosomes, which are organelles in the cell. They're designed to dissolve and break things down. They're very acidic. And so what we end up doing is by providing this acidic environment, accelerating the release of the metal ions into the rest of the cell, because that's accidentally sent this metal nanoparticles to the most acidic part of the cell. Okay, so skipping over that data, as I mentioned, probably time, we now know that endocytosis is important for uptake of toxicity. So I told you, what are they doing to cause toxicity? I've shown you some unhappy cells. Well, we thought maybe it's mitochondrial toxicity, and that was partly based on those TEM. It's partly based on a large number of papers in the literature saying that if you take silver nanoparticles and expose cells to them, you get loss of membrane potential and you get these various effects. We didn't find that very compelling because those tend to be effects that can be caused as a secondary result of lots of other kinds of toxicity, but they're interesting. What we thought was actually more exciting and compelling was that there might be an evolutionary reason to expect silver nanoparticles to affect mitochondria. And that's just the fact that mitochondria descend from bacteria, right? So endosymbiosis is how bacteria got into us and became our mitochondria. And we're using silver nanoparticles to kill bacteria, would they not also likely affect So it seemed like a very logical, great idea. One of several in my career that were probably mostly wrong, but a fun one to test nonetheless. So I'll walk you through some of those tests. Oh, and this is actually the point I want to make that's interesting. This theory has actually been borne out for some other things, not silver nanoparticles. So, for example, some of the antibiotics that we intentionally use, in particular some of the nucleosides and fluoroquinolones, target mitochondria. And some people actually got toxic effects as a result of exposure to those antibiotics precisely because they're targeting the same molecules in mitochondria that they target in bacteria. So it seemed like a reasonable idea. So to test this, we asked a number of questions. Do silver nanoparticles get to mitochondria? Are mitochondrial functions altered by silver nanoparticles? These last two I'll explain in just a moment. Are cells growing in lactose more sensitive than cells growing in glucose? That's a test for mitochondrial toxicity that I'll explain in a moment. And do genetic deficiencies in mitochondrial function sensitize cells? So first of all, do they go to mitochondria? This is work actually done in a mouse liver cell line, and the answer is not really. So these pictures are red cells. Blue is the nuclear DNA, so it's indicating the nucleus. The red is a dye for the mitochondria. So you see they form these sort of beautiful networks that go throughout the cell, and the white spots are the silver nanoparticles. And there's just not any, there's some that appear to overlap, but it's not very compelling. They're certainly not especially concentrated there. It's a little hard to tell with microscopy. These things are smaller than the resolution microscopes we're using. So there's a caveat there. So we also took a different approach where we took the cells and isolated cytosol or isolated mitochondria, et cetera, and then measured the silver content by Assay EMS. And those results are shown here. And again, we get a little bit in mitochondria, but it's mostly in the cytosol and cell debris. We don't have any evidence that the silver particles are especially concentrated at mitochondria. Of course, you can't prove the negative, right? So maybe there may be enough out there that that cause an effect, and we just don't know that. So the next test was a functional assay, and that was to ask whether cells growing in the lactose medium would be more sensitive than cells growing in the glucose medium. And the reason this works, this was actually a screen developed by Dunn at Pfizer to test potential mitochondrial toxicity of drugs. The reason this works is that cells that are in glucose actually don't really need a mitochondria. They can just do glycolysis and get along. And so if you poison their mitochondria, not that much happens. Whereas cells growing in the lactose really pretty much have to do oxidative phosphorylation. And so if you poison their mitochondria, the cells get really unhappy. And that's illustrated with this positive control chemical, FCCP. This is a mitochondrial uncoupler that dissipates the proton gradient in mitochondria. And you can see that the cells grown in lactose in the light gray are far more sensitive than the cells grown in glucose. So that's not telling us anything else. Nanoparticles, that's just showing the assay works. If we look at silver nanoparticles, there's not much difference. There might be a slight sensitivity in lactose, but it's very small compared to this. And we saw the same thing with some other silver nanoparticles, maybe some sensitivity, but not huge. And the same result also with silver nitrate. So that didn't really support this idea of mitochondria being a major target. Next thing we did was try to directly measure mitochondrial function. And this is using an oxygen consumption assay. So mitochondria in most cells use most of the oxygen in the cell. And you can measure their oxygen consumption just by watching oxygen concentrations go down in the dish. And so we did that at concentrations of different silver nanoparticles or silver nitrate that killed. So this is now LC50, lethal concentration, 10 or 30% of the cells. So sort of mild toxicity and then pretty significant toxicity. The idea is if loss of oxygen consumption. If the loss of mitochondrial function is driving the toxicity, we should see the loss of oxygen consumption occurring at lower concentrations than what it takes to kill the cells. And we don't see that. The cells are dying and there's still no real lack or decrease in oxygen consumption. Again, to me, this argues that mitochondria are not the major target of whatever silver nanoparticles are doing in the cell. Okay, so that was some cell culture work. Cell culture is not always a great model for what happens in full organisms. So we'll come back to our worm model and see what we can see in the worms. First thing we did again was ask about silver concentrations in the mitochondria in these worms. And so we exposed them either to no silver or the various silver particles or silver nitrate. And interestingly, some of them are taken up more than others. And these are at the same level of exposure in the medium. But if you compare the y-axes, we can detect the silver in the mitochondria, but it's not as high as it is in the whole worm. And so again, we just don't find evidence for strong concentration of silver in the mitochondria in the worms. However, when we did the oxygen consumption assay, and this is a more complex version of it, and I'm not going to walk you through the details, we're happy to talk about it if you're interested. We're looking at different aspects of mitochondrial function now. In all cases, we do see a decrease in oxygen consumption with these different particles. So that does suggest that mitochondrial oxygen consumption, well, shows that mitochondrial oxygen consumption is effective. How do we square away the lack of seeing silver in the mitochondria? Either it's an indirect effect, or we're just not able to see, or the bit of silver we're seeing in the mitochondria is enough. I lean towards thinking this is an indirect effect, and that's partly because of the last piece of data I'll show you, which is related to these genetic mutants. So, the concept of genetic sensitivity, I think, it's a useful one as a test. So, again, the idea is that if you have a mutant that's sensitive to mitochondrial dysfunction, and you expose it to something that might affect mitochondria that's more sensitive, that says that the mitochondrial function matters. But this also matters for human health, because we're all genetically different, and we are all more and less sensitive to certain exposures than each other. And so there are people in the population, for example, with DNA repair sensitivities, because they have deficient function in DNA repair, they can't go in the sun because they have a 2,000-fold increased risk of skin cancer if they do so. And so it's a major concern that we look out for people who might be genetically sensitive. So we decided to look at several different mitochondrial mutations involved in mitochondrial fusion, mitochondrial fission, and mitochondrial autophagy, also called mitophagy. And in case you're not familiar with these, I'm going to describe what these are briefly. This is showing you that mitochondria, and this is only a few minutes, fuse and divide very actively in cells. So if you look up here, these mitochondria are going to come together and fuse. If you look down here, these mitochondria are too slow, then they divide. And so this process of fusion and division going on all the time. Mitochondria are not kidney beans, and they're sort of these networks typically, and they're very active. They're constantly moving, very dynamic. And that matters for mitochondrial homeostasis for a number of reasons. But one is that as mitochondria divide, often one of the daughter mitochondria will be unhealthy if there has been damage. Because they're divided somewhat at random. One of them may be really pretty sick and the other one's fine. The sick one can be identified as such and sent off the lysosomes for recycling, basically. Break it down, make new, and then the fusion allows that cycle to go on. So this is this process of mitochondrial fusion, and then this breakdown of lysosomes is called mitophagy, are critical to maintaining the health of the whole population of mitochondria in cells. And there are Z genes. So there are mutations in these genes present in the human population, and they cause a variety of diseases. So outer mitochondrial membrane fusion is regulated in part by a gene called NFN2, mitofusin. And in roughly one person, 7,500, there was a deficiency causing a neuropathy. Intermembrane fusion causes a much more rare disease. The deficiency causes a much more rare disease called dominant optic atrophy. TRIP1, this is the fission, one of the fission regulators. It's actually very rare to find deficiencies. They do occur, they're quite rare. And then two of these mitophagy genes are called PARP and PINK1. They're so named because they're involved in Parkinson's disease, genetic Parkinson's disease. So they're also not terribly common, but they are the ones you may have heard of because of their role in these important diseases. So the question is, if you have a deficiency in these genes, would that make you more sensitive to a mitochondrial toxicant? The answer to that is yes. Will it make you more sensitive to silver nanoparticles, which might give us a clue about whether those particles are targeting mitochondria. And so we tested mutants in CLEMs that carry loss of function mutations in the worm homologs of all of these genes, which I'm just showing the names up here. And so this is what this data looks like. This is a subset of it. Here we're taking bone Arabic 25 particles and increasing the concentration. And we're comparing wild type, which is the gray size. So is the growth inhibited, and it's really not over that exposure range. What about worms with mutations in the mitophagy genes, the PARP and PINK genes? They're slightly more sensitive. Similarly, worms with mutations in the fusion genes are somewhat more sensitive. Their growth is arrested more by the silver particles. And we saw some more things observed on the fission gene. However, when we look at Arabic 6, which is actually more toxic than this one, we see only a small degree of increased sensitivity with the Parkin gene, the ER1. We actually see slight sensitivity with our fusion gene and resistance with our fusion genes. And maybe a tiny bit of sensitivity with the fission genes. I'm not going to walk you through all of this data for all the particles we tested because it's a lot. The take home was that there were some differences and there were some small sensitivities, smaller than we generally see with other chemicals we tested that are definitely bona fide mitochondrial toxicants. So we did, again, it suggested to us that mitochondrial function is probably important to support the cellular and organismal response to toxicity. You need energetics to respond to toxicity. But it doesn't really look to us like a primary target of the nanoparticles. Okay, so I'm going to actually wrap up here with just some conclusions. The first one, mitochondria are probably not the direct representative of silver nanoparticles, although they are probably important for allowing cells to resist that toxicity. Some people may be sensitive. Now, worms, of course, are not in fact people, but based on our data, it's a reasonable concern to wonder whether there might be some sensitivity. Most silver nanoparticles cause most of their toxicity via dissolution, but this potential for a particle to get somewhere that the ion by itself would not and then cause toxicity, sometimes referred to as the Trojan horse effect of nanoparticle toxicity, doesn't appear to be real, especially for particles, silver particles that dissolve more slowly. And back to the very beginning, most environmental factors that we looked at seemed to decrease the toxicity in a more ecological context. So, back to the big picture question, are silver nanoparticles good or bad for the environment? Health, and of course the answer, the answer for essentially every chemical you can think of is yes, they are both good and bad, depends on the context. I think what we learned enough to understand that there are some principles we can apply to the silver nanoparticles more broadly. The group I was part of, that's the point that out at the beginning, SAINT, the multi-university center for environmental applications in nanotechnology. There are some rules that have been identified and used in trying to understand toxicity. It is though important to actually use those rules and use those insights to protect ourselves. Right? So I would say these are not the most widely toxic things out there. Mark Wisner, who ran that center, used to give a talk at the end of our years saying nanoparticles not next XXs. But there are risks, and we have to be careful, and it would be smart to regulate it and be careful of those. I haven't given you yet the cardinal rule of toxicology. I would be remiss if I didn't, that those things look poison. This is from a clinical study of a person who bought colloidal silver, which you can buy at Whole Foods in the health section, and decided that a little was good, a lot was better, and drank a lot, and came down with acute psychosis, copper deficiency, and a variety of other effects, and was essentially cured by removing silver, probably part of her diet. So, yeah, again, there needs to be some care taken in terms of how we regulate and what sort of precautions are taken. But I think we've made some significant progress towards understanding that and thinking about what rules should we use to try to prioritize testing of different particles. I do want to point out a couple of caveats. Most of the studies that have been done so far have been more focused on shorter-term effects. There haven't been a lot of studies on things that take decades to occur in people, like degenerative disease or cancer. So I think that remains a question of concern. And although there has been work on environmental implications, I think there's definitely more room for that. People are more complicated than worms, are more complicated than cells, and ecosystems are way more complicated than people. So it would be nice if we had more opportunity to study potential ecological implications. That hasn't happened so far. With that, I'll finish, and thank you for your attention, and be happy to take any questions.
Is the specification of the silver always a silver +1, or is it there elemental silver 0, or is part of the nanoparticle?
There is, yes, there is elemental silver. In fact, particles are in significant part silver 0. However, on the surface that can change, and certainly when they dissolve, that changes. And that's silver, unlike, for example, people also look at silver at copper and iron nanoparticles. And there, I think there's stronger evidence for oxidative stress because the ions themselves cause oxidative stress because they can be upcycled. And silver, under physiological conditions, doesn't really seem to do that.
What are the main mechanisms of exposure that you're thinking about when it's something else? Is this from sort of environmental waters or acid streams or treatment?
Yeah, no, those are great questions. So one of the concerns was consumer products. So I have been thinking about things like band-aids and wound dressings where they're intentionally used to prevent bacterial growth. As it turns out, I don't understand what the intended benefit of drinking colloidal silver is, but people do it. So there is exposure that way. I think the wastewater concern is an interesting one because, again, most wastewater treatment plants have a stage where there's a microbial breakdown of stuff. And this could very likely interfere with that. That's certainly been a problem that's been seen in the past with other sorts of chemicals. Some of the pesticides in the past, when this is decades ago, when certain plants would manufacture pesticides and dump their waste in the sewage treatment plant, they would literally take out the function of those digesters. I don't know whether silver nanoparticles or silver ions released from them would get to a high enough concentration for that to be a major problem. I haven't seen data on whether that would get to a high enough level. Okay. Sorry, I don't know names.
I'm sorry. The oxidation state of silver, because you were using silver nitrate, which is the ion, and then I didn't understand when you were talking about in general silver nanoparticles. Are they metal state or are they also ions?
The particles themselves are mostly silver 0, except for maybe some of the surface.
So at one point, there was a consumer product that was, because silver particles tend to be antimicrobial, you have it in large volumes, and you get a spray bottle, and you spray your kitchen, you spray all over. And I don't know what the status of that, and the sheer volume was daunting. And from what you looked at, it would, and most of that would end up going into the sewage treatment plants, as well as possibly making certain things resistant type of thing. So do you know what the status of sort of the silver particles or silver nanoparticles are in the commercial arena?
They are certainly still being used for purposes like that. I don't know whether the volume, I don't know quite what the volume is, but you also raise another really interesting point. So there has been evidence. So one of the original hopes for these was that, of course, we're dealing with microbes that are becoming resistant to almost every antibiotic we have. And so one of the thoughts was that these silver nanoparticles could provide, because it's a different mechanism of killing, something that we could use that the bacteria are already resistant to. And that is true. It is a different mechanism, so something that's resistant to many of our organic antibiotics is not typically resistant to nanoparticles. However, nature is really good at evolution, and it turns out that bacteria, yes, indeed, do develop resistance to silver nanoparticles over time as well. I think it's still reasonable to think that if they were to be used in a reasonable way, they could be a nice part of the toolkit.
Well, I think the surprising thing about how they are immune, as you said, on bandages and the like. The scary part was large volumes in all of our hands being used sort of non-discriminately as an antiseptic, really. And what I think your work has done is really show that this solution is so important. And then once that happens, it maybe so much. That might encourage more of this other commercial production. I don't know what the regulations are. As far as I understand, the Toxic Substances Control Act regulates on the basis of an element and not on its particular status. So if you have a nanosilver versus a silver ion, there's no difference except that biologically there is. And so I don't know whether we kept pace with that or not.
I believe you're right that we have not. I was actually just told yesterday by a colleague that there are not currently regulations from EPA or FDA or OSHA for nanoparticle exposure. And that's part of why I finished on that note that yes, I would not say that these are horrifically toxic, but everything can be toxic if there's enough of it. And so it would be smart, and I think it would be a wise precaution to come up with some regulations.
Products like coffee that get sold a lot. Silver using copper nanoparticles for the same purpose. Are people looking at effects of environmental toxicology with copper as well, or perhaps you kind of into that this synergistic effects between different of nanoparticles?
That's a great question. So people are definitely looking also at copper nanoparticle toxicity. There's been little work on possible combinatorial effects of nanoparticles or nanoparticles in combination with other chemicals. There's been a little bit of that. And that turns out to be kind of an interesting literature because sometimes the particle absorbs whatever your other nasty chemical is and actually reduces its toxicity. Other times it sorbs it and becomes a vector for moving it into the organism, and that's obviously been a bad thing.
Put it in perspective. At this stage, it's remarkably good. Combinatorial effects of pharmacology is like it's not a lot.
some studies keep up with their actions. Some of that, it's actually a good sign.
That's a nice way to look at it. I unfortunately do agree with your perspective on how slow that has been to be addressed. I think it's a
The mitochondria, so where is your research going with the mitochondria?
well, so mitochondria is actually the major focus of my lab. Nanoparticles has been sort of one part of that, but in general, what we study are the effects of various sources, as well as other things, diet and exercise, for example, on mitochondrial function. Now that's sort of a whole different topic, but it turns out that chemicals, not all chemicals, but there are many chemicals that do affect mitochondria pretty dramatically. And it can do so in lots of different ways. It can interrupt electron transporting complex function. There are toxic chemicals, sort of like that FCCP example I gave, that work by uncoupling mitochondria fats. There are certain pesticides that work that way. There are others that redox cycle. Some that cause, this has been one of my lab's big areas of interest, cause mitochondrial DNA damage. So mitochondria have their own genome. It's very small compared to the nuclear genome, but it's there. And if you get mutations in it, that causes also quite a few human diseases. So that's been another big area of interest of ours.
Actually, that's new. There are a lot of toxicology studies in the mitochondria.
That's part of why I got into it. Now, I do want to say there certainly are some other groups doing it and doing a good job of it. But it's I got into it because I thought it needed more attention and because they're interesting.
Change the scale a little bit. When we're talking about fabrics which have the silver components in it to try and be antimicrobial so you can wear your t-shirt for a week, I don't know quite why we want to do this. We're not close on the scale to the nanoparticle size that we're talking about here, I would presume. Is that right? Or is nanoparticles simply a matter of they will appear in the same way microplastics appear because of breakdown and abrasion and so on. So I guess I'm trying to figure out what the threat of different sources.
Yeah, they actually, the ones put into clothing actually are often nanosized. So as far as I know, unlike the plastics where a lot of those are produced just by abrasion, this is actually intentionally what people manufacture.
Good.
Actually, the original first application of nanoparticles clothing or garments for space. Yeah. Well, can't take much longer now.
One point you mentioned, the sulfidation releases of toxicity effect. Is that mechanistically understood as an activation effect or?
I think it's mostly the particles end up being really well coated, and that ends up certainly reducing the dissolution rate of the particles. To the extent that particles have surface chemistry, and because this has been seen also especially for some other metal particles, it's probably also reducing some of that surface chemistry. To the extent there is dissolution, even if that's not prevented. Of course, sulfides bind metal cations really well and precipitate them out of solution, at which point they're not typically bioavailable and therefore not typically toxic. So that would be a secondary effect.
Is that generally true for a variety of compounds?
Yes. Yes, and in fact that's true for, again, this is sort of a rule that we learned a long time ago about just metals in general. So if you have metals in a sediment, let's say at the bottom of an estuary, and you ask whether those metals get into the food chain, They do to some extent in general, but the more sulfur there is in the sediments, the less gets out of the sediments. And that's been really well understood for metals in general for a while. It turns out to track with nanoparticles.
So when the nanoparticles are dissolving, what's the lifetime of the particle? So the toxicity is coming from this dissolving. Are we using up the particle in the time frame of this effect, or is most of the particle still there?
That's a great question. It's really variable, so some of them will dissolve really within a few days or at most a couple of weeks. Others will last quite a bit longer. It depends heavily on the environment, so a more acidic environment, of course, they dissolve quite a bit quickly. And then coatings can have a large effect as well. I'd advise against buying this silver imbued socks, though, because they're not going to remain silver imbued for all that long.
Thank you for a great lecture.
Thank you.
Thank you all for coming.
One of the things I came across earlier today is I keep